students. Today we are moving to the last part of the digestive gland, that is the intestinal gland. So you can see in the intestine region we are going to have two different types of glands. One is the crypts of liberkuhn and another is the Brunner's gland. So the first one, that is the crypts of liberkuhn, that is present in the region in between the two villi. We are going to have a depression, and in the depression we have the crypts. So those crypts, they are going to have the cells, intestinal cells. So those cells, they are called as the Paneth cell. I have written here, that is the Paneth cell. So this Paneth cell will release different types of enzymes. And this crypts of liberkuhn, that is present in the mucosa region, which is the innermost region of the small intestine. Now, if you see the second one, that is the Brunner's gland, just remember this Brunner's gland is important, and last day also I have told you that this Brunner's gland, that is actually the gland present in the region of the duodenum, which is present in the submucosa. So suppose this is the mucosa region. Here to this region we have this lumen, and if this is the mucosa, right, then here just beneath that we are going to have the submucosa. Okay, so in the submucosa region we are going to have the Brunner's gland, and the Brunner's gland, they will secrete a secretion directly into the lumen. So just see, remember that the crypts of liberkuhn, where it is present in between two villi, right, and that is present in the mucosa. I've written just M, mucosa, remember. And if it is a Brunner's gland, that is present in the duodenum, okay, in submucosa. Okay, so this is important, remember that. So as a whole, the intestine, which include the duodenum also, then the jejunum and the ileum, together it is the small intestine, right? So this small intestine will release different types of enzymes. So here the small intestine is also the region where almost all kinds of food will be digested. It will be converted into its simplest monomeric form, or suppose if it is a carbohydrate, it will be ultimately converted into glucose or the other monosaccharides in the small intestine. Protein ultimately will be digested, broken down into its simplest product, that is the amino acid. If it is a lipid, in the same small intestine it will be converted into the simplest, that is fatty acid and glycerol. And if it is the nucleic acid also, that is the chromosomes, DNA and RNA, eventually it will be digested even in the small intestine to its free bases, pentose sugar and a phosphate. So let's just see this small intestine. First of all, you can see this is the structure. In the small intestine, we are going to have this innermost region, that is mucosa. That mucosa is going to be just having some finger-like projections. Those finger-like projections are called as the villi. Inside the villi, we are going to have the lymphatic vessels, that is called as the lacteals. Remember, that is called as the lacteals, who will take the food inside it. They will absorb the food. And here we are going to have this arteries and veins towards the villi region. In the finger-like projections, it will be divided into even more fine branches. They are called as the capillaries. So from the capillaries, the lacteals will absorb the simplest food. Here in this region, what we can see, these cells that will form the crypts, that are called as the crypts of liberkuhn. So this is the diagram. You need to know this is actually the section of the small intestine where villi are present, and inside the villi, the lymphatic vessels are called as the lacteals, who will absorb the food materials, and it will directly come back to the liver to give that food material to store inside the liver. Now just see this intestinal juice, which is actually released by the crypts of liberkuhn and the Brunner's gland as a whole. In the small intestine, the juice basically is basic in nature. In the in the stomach region, the food that is mixing with hydrochloric acid, so that region, stomach region, the food is acidic. The juice that is releasing, or that environment, is acidic, where basically the proteins are being digested. In the small intestine, this juice that is basically basic in nature because of the bile, and bile as a whole in the region of the small intestine makes the food basic, so that the leftover food particles that can be digested into its simplest form. Now just see, in the small intestine daily we are releasing around 1 liter of intestinal juice per day, and it is alkaline in nature. Remember, this alkalinity will be varying from 7.3 to 8.3, right? Remember that. Then the cells and their secretion. See, in this small intestine we are going to have different cells, not only the cells which is releasing the enzymes but other cells also, like goblet cells. So goblet cells have to be present everywhere because it is releasing the mucus, and mucus makes the food slippery so that we can able to properly deglute, that means we can take, or the food can pass through the small intestine smoothly. So that's why goblet cells will be present, and it will be present not only in the small intestine but throughout the GI tract or the alimentary canal. Now next one, there is a cell which is argentafin cell. So this argentafin cell I have told you in the stomach region also, which is releasing serotonin, and the function of serotonin, we know that it is a vasoconstrictor. So this serotonin will squeeze so that the secretion from the cells can be released. Next one, there is a cell that is also present in the stomach region that is called as the basal cell or the stem cell, which will regenerate any of the degenerated cell. So if any of the cells they have been degenerated, then the basal cell will regenerate those cells once again. Now here the main basic cell which is releasing the enzymes, they are called as the Paneth cell, right? And also one thing we need to know, that here in the small intestine we are going to have the Peyer's patches. Okay, so this is called as the Peyer's patches, that is actually the lymphoid tissue. Okay, so this lymphoid tissue will basically help us to kill the foreign particles if it is present along with the food. So it is protective in nature, that this Peyer's patches will kill the germs. Like in the region of the pharynx we are going to have the Welder's ring. Similarly in the small intestine we are going to have the Peyer's patches, which is actually a lymphoid tissue which will kill or protect us from any foreign particles or pathogens. Now come to the Paneth cell. So this Paneth cell, which will have different types of enzymes, and different type of enzymes will act differently. That means that will act on different types of food, which will eventually break the different types of food into its simplest particle. So first of all, what is that? That is enterokinase. Remember, this enterokinase will have one function, that it will convert one enzyme, that is pepsinogen, which is in the inactivated state, to pepsin, and that pepsin will work on the protein and convert into the simplest food particle, that is the amino acid. Next one, another enzyme that is called as the erepsin. So this erepsin will convert the peptide into the dipeptides and the amino acid. Next one, another enzyme is your dipeptidase. So first of all, erepsin will work on the peptide, which will break the peptide into dipeptide. So what is peptide, what is dipeptide, everything we will understand whenever we will discuss biomolecule chapter. Just for now you just remember this way, and whenever we will discuss biomolecule we will join and we will understand all of this together. So first here, this is the dipeptidase that will break the dipeptide into the peptides, and peptides that will break the peptide, that is dipeptide, into the amino acid. Actually two amino acid will join by peptide bond and it will form the dipeptide. Dipeptide with another amino acid will form the tripeptide. So this way the peptide bond will join the amino acid. So the two amino acids, if it is attached by the peptide bond, this dipeptidase will break this peptide bond and convert it into the amino acids. Next one, there is another enzyme present that is called as the lipase, which is also secreted by this small intestine, that will break the fat molecule into fatty acid and glycerol. Next one, maltase, sucrase and lactase. Remember, that will work on the carbohydrate. So this maltase will work on the maltose and it will convert into glucose. Next one, sucrase, it will work on sucrase enzyme, which will act on the sucrose and convert it into glucose and fructose. Next one, lactase, which will work on the lactose and which will break into glucose and galactose. So all of this will work on the disaccharides, and it will be converted into the simplest monosaccharides, that is glucose, fructose and galactose. Other than that, two enzymes are also present. They are the nucleotidase and nucleosidase. So DNA and RNA that will be broken down into the nucleotides. That nucleotides will be acted upon by the nucleotidase enzyme and it will be converted into nucleosidase. Then this nucleosidase, which is present as an enzyme released by the small intestine, that will act on the nucleoside and it will be converted into its simplest free bases. So this way the genetic material DNA and RNA that will be also broken down into its simplest form, that is the free bases. If it is a carbohydrate, maltase, sucrase and lactase that will act on the disaccharides and convert into its monosaccharide. If it is the protein, then yes, enterokinase, erepsin, dipeptidase that will work on the protein or peptides, polypeptides, and convert it into the amino acid, which is the simplest form of the protein. And if it is the lipid, this is the fats and oil, which will be acted upon by the lipase enzyme and eventually it will be broken down into fatty acid and glycerol. So that's why here in the small intestine, all the four types of food, that is carbohydrate, protein, lipid and nucleic acid, which will be converted into its simplest form. So yeah, that is all about the glands of the intestine. So we have completed the enzyme part. They're releasing from all the parts of the our alimentary canal, that is GI tract or gastrointestinal tract. First of all, we have discussed the mouth. Here we have the saliva. Saliva is released by the salivary glands. Then after that we are moving to the esophagus. Esophagus will not have its own cells to release its enzyme. So the food, whatever it's mixed with the saliva, that will go and continue on digestion in the region of the esophagus. Then we have discussed the stomach. In the stomach, we are completing the different types of cells, and those cells that will release different type of enzymes. Basically one enzyme is here, that is the pepsin, and the other type of cells are also here, which will release the different type of other things like hydrochloric acid, casein intrinsic factor, then serotonin. This type of cells we also have discussed. Then after that, small intestine. Here small intestine will have Brunner's gland, which is present in the submucosa of duodenum, and the crypts of liberkuhn, which is present in the jejunum and ileum, which will release all those enzymes which will convert all the types of food products to its simplest form, that is carbohydrate to the monosaccharide, protein to amino acid, fat to fatty acid and glycerol, and nucleic acid to the free bases. So that's all about the different type of enzyme. We have completed all of the enzymes. In the next video, we will start discussing the different types of food, that how it will be digested, in which region it will be digested. So that's all about the parts we'll discuss in the next video. So that's all about the video. I hope you understand. Thank you.